Well, hello, everyone. It's really wonderful to see you all. Thank you so much for coming to the Lab Futures Forum. My name is Daniel. I'm one of the product directors here at Automata, and it's my pleasure to host tonight's event. Thank you all for joining us. It's great to see a few familiar faces and indeed a few new faces, so welcome. As many of you may know, but some of you may not know, Automata's mission is to make lab automation a reality for every lab. And to that end, we developed the LINQ platform that we've all been looking at here tonight on our lovely sixth floor demo space with beautiful views over London. And this platform really represents our committed vision to the future of lab automation. And through its open, modular, scalable design and its integrated cloud-first software suite, this platform makes end-to-end workflow automation a reality for almost every lab. And it was during the design and development of this platform that we started the Lab Futures Forum. And we started this forum as a means of progressing the discourse around technology and its applications within life sciences labs. And we did this to keep us on our toes and indeed to keep those that share our passion on their toes about exactly where this industry is headed and why, staying at the forefront of the conversation. Coming up next month, I have to give a shameless plug here. We're going to be hosting Tomasz Rzeka of Artigen, an AI CRO, who's going to be talking about lab-in-the-loop applications. And in December, for our final event of the year, we'll be doing a Christmas social in conjunction with the Lab Automators, London Lab Automators group. So that'll be fun, and do please join us for those events as well. Stay tuned and pay attention to the events page on Automata's website for more details. But for tonight, we're going to kick off with my colleague, Russ Green, here on my left. Russ will be speaking about automation, the validation steps that it provides, and how automation enables the data factory needed to drive in-silico design. And following Russ, we'll have tonight's esteemed guest speaker, David Wright of Kuano, who certainly delivers on the goals of the Lab Futures Forum. David will be speaking about his work on in-silico drug design with quantum and AI. After we hear from Russ, we'll be taking Q&A from the audience, both in person and those of us joining online. So do please prepare your questions as the talk carries on, and we'll be sure and answer your questions at the end of the evening. So I encourage everyone to give a big round of applause to Russ Green. Russ.
Hi, thanks everyone. So before David gets up and talks and blows us away with some amazing applications of AI and physics in ways I don't fully understand to advanced drug discovery, I want to talk a little bit about some of the role that automation has to play in driving some of these lab in the loop or AI-driven applications. And I want to speak explicitly about how we build data engines. And what do I mean by a data engine? Well, fundamentally, we talk about an automation platform, but it's more than that. It's an automation platform that drives collection and structuring and harvesting of highly contextualized data. I don't think there's been a single client we've spoken to who's trying to advance their science in this space who hasn't talked about data, and specifically quantity and quality of data, being one of the fundamental problems that they have. So when we think about how we bridge the gap between what we have today and how we might utilize AI at the bench, there are a few pillars that we want to think about. One of them is about how we might apply modern resourcing, so cloud compute technology and modern cloud infrastructure. Another aspect we have to think about really hard is how we actually drive that wet lab integration. So how we might use that modern stack to dive deep into what we're doing in the wet lab space. And then at the top of this pyramid is the thing that I mostly want to touch on today, which is around data generation. And so I don't just mean about generating results, which is what many automated systems have done before. I mean about generating high quality, highly reproducible, highly contextualized data and what it takes to get to that. And this is a journey that we're going on with quite a number of our clients at the moment. And it always seems to take the form of a number of specific steps to get there. So when we think about this and how we build this data engine, the first step we need to take is actually kind of an obvious one. It sort of fits in with how we build most automation systems, but it's how we can go from the concept of this bit of hardware that sits in the middle, like I haven't got my pointer working, to then thinking about that full data pipeline, automation pipeline. So we need to go through the process of taking a system that can take experimental designs, run and gather that data right through to capturing results, and then merging that data back with the experimental designs and placing it into a location that is readily accessible, findable, and structured for whatever other part of the ecosystem needs to consume that. Now, getting this bit working isn't that straightforward. It should be easy, right? Setting up a system just to gather data. But actually what we find is that this leans really heavily on people like automation specialists to drive the optimization of these systems. I'm going to speak very briefly about what that looks like in their world and what we need to think about as an industry as we're trying to enable this. So a good lab automation ecosystem should be open, right? It should break down some barriers to enable different kind of people to be able to interact with different parts of the platform. And it should also open up the export of that data and how we get that into the ecosystem. So speaking explicitly about how we build workflows and optimize for them, there are different kinds of people we have to cater for. So we might need to think about a code savvy user or a non-code savvy user and enable completely different journeys to get to something which fundamentally at the end should be the same. We should be able to develop workflows that are robust and executable in a routine way. So we can then think about what that looks like when we go through that optimize and validation process, where we take that fundamental process we built out and then build it out into something which is going to either maybe train our AI model or even just optimize for a particular kind of experimental scenarios. So in this example here, we've now inserted a human into the loop, really to analyze that data and retrain it. And we've seen a couple of really good examples of this where we're trying to train up ML models. And so what we're doing is using the human to interpret data that's coming off the platform and deliberately redesign an experiment to go back into that core platform to develop another set of data that will feed back into that ML model for training and go through that iteratively to try and build out that training model for that with that huge data set. Once we've been through this process, we can then start thinking about closing the loop. And this is where the openness of the platform, where the world we tend to live in tends to sit up here, and we need to be extremely open at one end to be allowed data to be pulled or pushed into whatever part of the ecosystem is required. And at the other end, accept new experimental designs that might have come from having analyzed that hypothesis against a particular scenario and then iterating on perhaps an experimental design. So with that, I'll just close by sort of describing the kind of closed loop we're trying to look at and enabling through a much more open architecture. We can think about this broken down into the science of a system, the work cell we're collecting data on, and then the data itself. Where we go through that process of building workflows is typically where we start out in experimental design and then translating that into automation. Executing upon that system, on that workflow to gather huge amounts of data, contextualizing and structuring it and placing it into accessible places in the ecosystem, and then pulling or pushing it to whatever that client might be afterwards. So that could be the scientist or a human user. That might be another bit of cloud-based infrastructure or perhaps our AI or ML, which can then update workflows and interpret new data and redesign that workflow to iterate on that cycle. So just some thoughts about where we're heading to and what we're seeing an awful lot of demand for about how we have to really focus on robustness of these data engines to be able to drive this closed loop and both input new experimental instructions and output highly contextualized data. And with that, I'll stop talking. I'm going to hand over to David to speak about some of the really exciting stuff they're doing at Kuano. Hopefully that should work. Yeah.
So, really a pleasure to be here today, and just sort of slight warning that this is a more opinionated talk, and I've been doing it for a while, so maybe. Sure. Yeah. Thank you so one way that was sent me by from your company was the goal of all of the discovery is to go from the cell paper to the clinic. Like you're going to make them innovative in scientific sense. Do that one, so you can make different. Truth of that in like truth of these figures at the bottom of the thing sort of argue apart from the paper, right? Fails all the time. Yeah, but it costs a lot of money and it takes a long time. So people really want technology somehow prove that journey. And one of the ways that people think about that is like, should we transform these messy, horrible problems to biology into simple engineering problems a bit. Dynamics and other tools help you build bridges, build bridges, parts, optimize parts. Is this a challenge that you can do and where the so recently,
Apologies, David.
all right, yeah.
The people online.
So the two technologies that people talk about a lot in this space are can AI or quantum computing figure that in some way. And the rest of this talk will go through some of my answers to this. And some of them, if everyone in my company will agree with, everyone in the room will agree with. It won't. That'll be fun to discover where that is. So you'll read a lot of papers that sort of claim, make it sound like AI will solve all of the world. So I think that like it's really worth saying it does solve a lot of problems. It's pretty useful. This picture I've got here, I couldn't draw it myself. It would take you a million years to make a drawing program. I mean, even half of it.
Yeah, because David, a little closer to the mic. Thank you. There we go. Thank you.
Is that okay? At a reasonable bit closer? Perfect. So like Atomwise is a company, a US company who do AI, and like they've managed to get a system that really does sort of genuinely seem to do improving the in-silico, the hit rate of, when you go to high throughput things, but it's 73% to 50%. Is that amazing? It's pretty good, really, to be honest, compared with most things you try and optimize. But then that's taken $60 billion of investment globally to get to this sort of relatively modest improvement. And no drugs have yet gone into clinical trials from AI. And so you could reasonably say that we have the same failure rate at the important variable of this whole system. We have a 90% failure rate, but there are upsides. We do much better at phase 1 clinical trials. There are reasons for this, I suspect, but there is also really worth saying that like it is still helping us in various stages of this process. And that a lot of the hype comes from like these big failures, not from a global failure, but just because people have heard of 2 failures. And so this is kind of, what is the problem? Why is it not the same as like the AI search engines? And the argument from my position would be that like, one, it's just a different problem. You can look here and see I put in 2 relatively similar prompts to Microsoft Copilot. And I've got quite different sort of responses, both in terms of trying to make it like a Dixit card. I don't know if any of you know the game, but it's a game where you have kind of quite French artistic pictures and you have to sort of tell a story with them. One of them, I think, does a pretty good job. The other one rips off the cover from Codenames, a different board game, for no particularly apparent reason, right? That's not scientifically useful. These are things that are unpredictable and weird and wouldn't help me make decisions if I was doing something equivalent in a lab, right? And that's part of the problem here. But the other problem is there's just less data. Like how many molecules would any company test? How much would we get? Not the millions of things that you get, not the corpus of hundreds of millions of words, trillions of words that you go into your ChatGPT. This is a real issue that we just don't have this sort of stuff and we can't do it. And that's compounded by the fact that like representing things in a computer is also hard. Images are easy because there's an obvious meaning of up. That's not like a molecule in the world. Every configuration is different. It's just a different space. The conformation is different. The way that things link up, it's not even clear that the things that we think of as humans in terms of the network joining atoms together, which we would call bonds, do they really exist? Open actual question depending on how you look at this, and actually has an impact upon the decisions you want to make of whether this is a good drug for this target. And there's something worse than that, I would argue, which is that if I have a black box model, I can't explain it to the people who have to do the really hard work of doing an experiment to try and work out whether or not my nonsense is, real, is actually a surprising and non-intuitive answer, or is it just a set of random bits that I've made into a pretty picture somehow? And one of the ways that I think, so I did a PhD under someone called Peter Coveney at UCL, and he's very, very fond of saying that like all of this data-driven work has its obvious limitations in having no understanding, no physical model around it. And quite a lot of the rest of this talk, certainly the bit about the company I run, Kuano, is about how can we unite this type of really incredible imaginative technology with physical models that ground it with a reality that can be used to make decisions in drug discovery. So that's physical models helping us out of this problem of sort of madness. The other part of this is about a more obvious thing, or more obviously linked to physical problems. Like, so for a long time, we've realized it takes a really long time to do anything useful with a computer on chemistry. Like, so I used to do a lot of molecular dynamics, and this meant weeks of running a thing to wiggle it a little bit to find out not very much information. Potentially, quantum computing offers us this magical world whereby the computer in some way replicates the structure of the problem and therefore gives us an incredibly precise answer for almost no effort, right, in some way. The slight problem with that being that we have no quantum computers to do quantum computing with it, but we won't dwell too long on that because that's the next slide. But there's been a lot of talk for a long time about an idea of quantum supremacy. And what does that mean? It means doing a problem that you couldn't do on a classical computer easily on a quantum computer. But the important part of that people sort of seem to miss is that it's irrespective of the usefulness of that problem. I'm not super interested in what, say, the Google claim of quantum supremacy was, which is how easy it is to train a network to do quantum computing on a quantum computer. Does that help me? Not often, right? Like, it's not a problem that occurs to me that often. Whereas the problem of what does this piece of chemistry do? What does it look like in some scenario? Can I just make the properties of that? That's a really good question that I would like to have answered. And so this seductive idea, so like with the seduction of having a answer from an AI model, but no understanding, the idea that a probabilistic computer can answer my probabilistic question is seductive, but it's hard because the problems I can answer it, but are not obviously useful. You might, however, though, go, but it is a computer that is based upon the same quantum properties. That's how quantum chemistry works. That's how chemistry works, right? So there is a neat link. And there aren't any pretty pictures, but I'm going to try and convince you that there are outputs from this that are still useful. So one of the questions you always get asked when you talk about quantum computing is, when will there be a quantum computer such that anything I say is relevant? And this is a paper from last year, which breaks down the space in what they call noisy intermediate scale the quantum era. So I moved from computational chemistry, which is all about 3-letter acronyms, into the magical world of 4-letter acronyms with this work. And the important point here is that most quantum computers made to date are roughly equivalent to a laptop in my sort of useful problem solving space. And most of the other ones are too noisy to do lots of useful calculations. So they come under the useless part of this bag, as far as I'm concerned. And this mythical green zone over here is where we'd like to be able to do something. But we have computers that are sort of edging in that direction, which is great. And like, one of the questions is, like, so, I mean, I phrase it as, is this another nuclear fusion scenario? And I really genuinely think it's not. I think that the hardware is showing genuine progress now. Niche problems will probably be solved in the next 5 years. And you can sort of see that we're hitting the hockey stick in terms of number of usable quantum bits. So it's not totally stupid to start thinking about what would we do once we have a quantum computer. An analogy I'd draw for those who've done sort of work in physical sciences is that GPUs came along, and there was a really long time when the real problem with using GPUs was that no one knew how to put any software to make use of that. And that's a real problem which only really was solved because AI became a big thing and people started investing in the infrastructure to use the hardware. But that's not the actual argument I want to make. I want to say that actually what's really interesting from a quantum chemist perspective is that we haven't really changed fundamentally how we do quantum chemistry on computers, other than occasionally using accelerator hardware, for a quite long time. The problems are solved the same way. And what I think of the analogy is that the quantum computing realm is like the Formula One sort of driving cars to the road car, or in this case, to the bus, right? So Williams, when there was going to be a tweak to the regulations, created an energy recovery system, which is now not used in Formula One at all, but is used in buses, right? A very day-to-day mundane application of incredible technology. And I think that's where in quantum computing, we can start reconceptualizing the chemical problems we have today to fit them onto quantum computers. But the use of doing that is not to run it on a quantum computer, is that I've tried to understand chemistry better. And so this is like something that we've done in our company, largely through grant funding, and it has 2 applications that actually we use sort of every day. I haven't really got into what we do, but one of the things is we try and take an enzyme, which is a comparatively big thing compared with a drug, and work out and run simulations to work out how well it binds to a drug. And the problem in some ways is that in some simulations, what are the important parts? If I want to put extra detail into my simulation only on those parts that adds value to my decision-making process, how do I do that? And one of the key things about trying to get a big system onto a quantum computer is how do I squish it down into the smallest amounts of parts that I want to do? So we've been working really hard on a variety of selection criteria to work out on the active space, which means the sort of orbitals that do some chemistry, and selecting it. And that's what we've found useful. So we run a simulation of an enzyme, say, through its reaction, but we only want to pay quantum money on the bits which are involved in the reaction. And we need something before that where we can get that. And that's exactly the same system that we would use to get a quantum problem onto a quantum computer. The other thing that's cool is that the reason why people think quantum computers would be exciting is that you can tackle harder and harder and harder and harder systems, right? And so the system that everyone uses to benchmark their quantum chemistry code in their realm of trying to do really hard things, even if they're not useful, is things with lots of iron atoms. And this is called FOMOCO, for no use to anybody here in the room, probably, but like it has a bunch of iron atoms, which everyone goes, don't touch them, they'll destroy everything. So this is sort of molecules you might see in hemoglobin or something where it might be useful, but you won't do anything with it because your quantum chemistry codes will fall over and die. Whereas actually what we find in this thing, which has 7 things which we wouldn't touch, is actually it's much easier than a lot of other systems that we would have touched. And what's important for us is that knowing which systems we should invest our money in and try and simulate and try and do drug discovery, that's a hard problem already. And now we have a tool that helps us to work out which ones our computational codes might work on that other people would throw away. So that gives us a competitive advantage. And so I guess this is where I come to essentially the kind of what is Kuano's dream and why do the things I've told you about matter to us at all, part of the talk. And so I'm going to talk you through a primer of the sort of fundamental theory we try and use to give us a different angle onto drug discovery, and then try and talk to you about how the technology I've sort of described impact upon our process and how the outcomes have come and how we validate in that wet lab. So I don't know how many of you are biochemists, chemists, or physicists, but basically, if I'm teaching you to suck eggs, it's not that long of your life. So the idea is that an enzyme, which is a common target for drugs, is a sort of little factory for chemistry. It catalyzes reactions, makes them go faster. And the way it does that is it takes them through the energy landscape, reducing the highest barrier to increase that rate, but it does that by binding incredibly tightly to these things. And so the idea here is I have initial sort of static state of it bound to its substrate, going through this transition state to the output of the reaction. What I'm interested in is this transition state, but I can't see that in a crystal structure. I can't see this in an MR structure. I have to try and do the chemistry that gets me from point A to point B. And so that's me having wasted a slide. So the advantage of being able to do this is that, as I said, it's incredibly tightly bound. That's the sort of thing I want in a drug, right? So if I could mimic this transition state, that would be great. That's high potency, a drug good at sticking to its target. But it's also incredibly sensitive to the entirety of the environment of the enzyme. So if an enzyme is doing it in a different environment, if it's built to do it in a different way, it will have evolved to use a different transition state. That's another property I want in a drug. I want to be able to hit just the one target, not all of the targets, right? So this is what we'd call selectivity. So selecting for my particular target. And that will reduce toxicity in the end. And then you also have this added advantage that because this is all based on the mechanism of the enzyme, it has to work harder to evolve resistance to this type of drug. And what we really want to be able to do is to take all of these features, find some non-intuitive chemistry that looks like them, but could look like a drug as well, meaning it could get through the body without being chewed up by things, and it gets the right part of your body to do its job. And not turn off the screen. So what we've built is a sort of engine full of a bunch of different technologies put together. And so this is just a thing for me to talk to say that we have a simulation engine that tries to run through reactions, and we need to have at least a quantum degree of accuracy in that part where the reaction happens. We can accept the rest of the enzyme is at a lower resolution, but we really need to have a high resolution in some areas of that simulation that captures quantum chemistry. We need something we call a pharmacophore. So for those who in drug discovery are familiar, meaning some sort of pattern matching scheme that allows me to say what properties I'm interested in and match them between two different chemistry and two different chemicals. And we want to have that to be bespoke and relevant to the type of high energy chemistry and also the stable chemistry. So that's the transition state is the high energy chemistry, and then the drug-like chemistry is the stable chemistry. And then I want to be able to have as imaginative a set of chemistry put through that filter as possible. So we need some generative AI at the. And so this is a quick case study where we've taken that system that I've just described to you and run it for drug discovery and shown that it works pretty effectively. And we're now on that process to like doing greater and greater levels of validation. And one day we'll have done it at a scale such that you believe me, I suspect. So the idea here is that we've taken this enzyme called NOTEM, and it takes in a substrate and it changes conformation in order to perform the reaction it wants to do. So we ran the simulation, found some really distinctive changes. Called an aptenion hole. What it means is it's redistributed the charge in the active site. It's changed conformation of both substrate, which is the thing we're trying to match, and itself. So that means the set of interactions that are available to that are different. And so what we do is we built a format for this sort of pattern matching thing. And that includes all of those features that we see in this unique structure. So it's different from what you see in your crystal structure, and it is druggable in the sense that it gives you features that you could put into a drug. And then we use the generative AI and we run it through. And so what was really exciting in our first set of validation is that we were working with Alzheimer's Research UK, who had done an enormous amount of screening and then not told us about it. And what we did is we found their best hit in our first run of this system from A to B. So that's the sort of type of blind validation of our system, in that we're finding the type of interactions that work well in the lab, and that actually, if you look at what we came out of our simulation and we matched our drug, it looks like what they found in their crystal structure. So they've done an experiment which sort of retro validates all of our work. What is even nicer than that is that when we kept going and decided that we'd do the much more lucrative cancer drug design project on the same target, is that we rapidly found two new chemotypes, so new types of chemistry that worked. And they not only did the right thing in the early assays, but actually they work really well in biology. So they've gone right the way through hitting the target we want, selectively hitting the target we want, and impacting the biology pathways we want in cancer cells, and actually cancer organoids, and they don't kill mice. So we have done all the things we're sort of promising. And so I guess that's really the end of my story of how Kuano has sort of validated ourselves by going from sort of this dreaming scenario where our computer does whatever it wants, to actually proving that it works in the wet lab, and that we've used in that process all the technology I've talked about. So the quantum computing, simulation preparation techniques, finding what's important, putting that through as a scoring function in our generative AI, and then using the generative AI to dream up new, novel, patentable compounds that work. And so I guess I would like to have a balanced view. It's a pretty exciting time to be working in these fields. And I think that we're not the only company, but people are now really demonstrating you can have benefits from AI, from simulation technologies. They are not magic, and biology remains messy and wet, and also complicated with many variables that are hard to control. But what I think is really interesting about having this time is that it's the time to rethink about your problems. Like, how do you pose the questions that you pose? How do you try and solve them and fit them onto a computer efficiently? And what type of answers are you willing to accept and take to the wet lab, right? What are pushing that science forward? And another thing that I sort of maybe have underplayed here is that in order to convince somebody who understands how to make chemistry to make that chemistry for you, you have to start interpreting what you've done. And a key part of this is we're slightly lucky that structural biology gives you pictures, but actually we've found it like the hardest lift is probably to get everyone on board and actually to explain the science we're doing. And that's like the challenge that we're facing now. And I think a lot of people are facing, is how to not make it look like you're trying to be a wizard and start trying to make decisions in a rational and clear-headed manner. And that is the end of my talk. And I just want to point out how beautiful it is that the slides here have the type of typos you'd really hope to have in an AI-generated image to end your talk about the dangers of just trusting AI blindly. So thank you very much for your time. It's been a real pleasure.
Excellent. Thank you, David. Well, I hope everyone enjoyed the talk, and I hope you've been formulating your challenging questions during that talk. We're joined here as well. I failed to introduce Tim Katte here. Tim is one of David's colleagues at Kuano, and will be helping to field some of the questions tonight. So David, thank you very much. There were some spicy comments there, and I'm sure you'll have a letter from Google and IBM in the mail very shortly. But I really appreciate your detailing this for us. You operate in a very rarefied air. There are not a lot of people that do this, and I think you've explained it quite clearly. And one of the things I love about what you described most is it creates a kind of adjacent perspective to all of the AI conversation that's going on, which helps to let us take a step back, step to the side, and get a little bit more perspective on the challenges there. One of the things that I really found fascinating was right at the end when you were talking about your application with was it the Alzheimer's Foundation, Cancer Research UK?
AI UK. So
AI UK. Thank you. And I was a little curious if you could expand upon that a little bit more. You said very quickly what was the sort of duration of that project? Can you tell us a little bit more about the character of that project? And also, can you tell us a little bit about the models you were working with, particularly on the AI side? Are these Kuano proprietary models? Are these models that are being brought to you by partners?
Yeah. So in terms of the project, so we were basically fishing for people who had the biology knowledge and the target knowledge that we wanted, and were sort of interested in working with a company who didn't have a big reputation. And so the Alzheimer's Research UK Drug Discovery Unit at UCL, which is fun mouthful, they had this project where they felt that they were struggling to engineer the sort of bioavailability properties into their target, and they were interested in finding new starting points. But they were looking to publish their data, so they weren't willing to share any of their data. But there was a crystal structure of the beginning, the thing. And so within, I think it was 2 months, we had a model run of the whole system, and we'd done our generative approach, and we had our initial set of things, and we're just sort of filtering them through. And I guess, within certainly beginning to end, 4 months, having everything synthesized, pushed to them to be tested, we had reproduced their in-silico thing and found things which did turn out to be 3 hits. So that was a useful validation. And I think you sort of asked about the models as well. And there's kind of a massive amalgam. We started very much with a custom version of, well, actually, so everyone will be familiar with sort of machine learning type models, but actually went back to genetic algorithms to start off with to give us a much bigger palette to work on and much easier. But we found the biggest problem with that type of modeling is really getting synthesizable molecules. We have really good ideas. It helped us to really explore our sort of physics-based models, but they weren't going to be made by any medicinal, by our medicinal chemists. We're going to say no to them, and then the synthetic chemists, we're going to say no to them. So we've then sort of adopted a lot of, they're not quite off the shelf, but academia adjacent models, which are based upon reaction pathways to make sure that we're getting more molecules.
That was amazing. And I imagine it's early days to answer this question, but comparing the sort of pace of discovery that you're seeing with more traditional processes, is it too early to really make that comparison?
Yes and no. So the more and more businessy I feel, the more I'm inclined to say things like, so Accenture take their benchmark as 2,500 molecules to something that can get into their in vivo modeling process. And we managed to get ours into that same process in 250. That's pretty good going. So it shows that certainly the potential is there. And I guess really the reason why on that sort of slide I showed the bigger picture of you don't want to get to your in vivo proof of concept, you want to get it to the clinic. And in that way, the real judgment comes when you've had your 5 to 10 years to 10 to 15 years, because you interact with the real world, right? Doesn't matter how fast we run, if the regulation slows you down 10 years, you're 10 years from the clinic, right? But that is something that's managed, there's manageable and changes, and there's different routes that give you faster routes.
Yeah. Earlier in the forum, Russ talked a little bit about the data engines that laboratory automation helped to propel. And you talked a little bit about the nature of AI, that it's really driving based on inference and data sets of the past, and that there really is a kind of lack in many cases of the data quality or quantity that's needed. And it sounds almost as though you're providing a bit of a cheat code here for low data environments. Is that a fair statement?
I think that's true, but I think when you think of it as a cheat code, it's like we're trying to take some of the pinnacles of human achievement of understanding chemistry and quantum physics and using that as the cheat code. A lot of effort went into that. It's not like it came for free, and it's not like the modeling doesn't cost anything computationally or in terms of the intellect of doing it well. I think it is true though that like if you think about using a machine learning model without some of that extra context, without the physics-based background, I mean, from that regard, purely from improving the performance of your machine learning model, I think yes.
So just to quickly interrupt there, sorry.
Sure.
We do have two questions from an online person. Stephen Zimmer is asking, why should AIDD be any better at finding effective drugs if the paradigm is the same target-centric one?
I mean, that depends. I mean, like, yes, there are just different problems here, right? And I think that the questions both, are the set of targets you interact with limited by your understanding of the biology or your willingness or knowledge of how to target that biology? And the answer is both, right? So having a more imaginative way of doing one of those things, in this sense, the chemistry part of that problem, tackling new targets. For example, people talk a lot about protacs or molecular glues as new modalities or covalent binders. If you have new ways to do that, and I think that's the sort of I mean, not AI, but different new technologies in this sort of computational drug design space are opening up that. And I think you also have people who are working on the biology, how do you find new targets problem, but that's not my specialty. Leave that to them.
Okay, I also have one more question from Stephen, and it's, do you believe that you can eliminate the inherent polypharmacology of drugs?
Again, I'll go back to my statement. I don't believe we're wizards. I don't think we should pretend to be wizards. So do I? And I think sometimes, depending on how you're defining that, whether if you mean a drug hits more than one target, that's sometimes a benefit, right? And I don't think we should necessarily take the serendipity as a bad thing. We need to understand what we're engineering and why. And that's, again, I mean, to your point about automated experiments, and linking this up to experiments that answer better questions as well is the only way forward.
Hi, thanks. Great talk. My name is Vikram. I've got a PhD in genetics and more recently a life science PC. Question. So you mentioned about target being druggable and identifying targets, but what about efficacy and things like receptor desensitization? How do you factor that into your model? Once it's bound, what happens? How do you predict that?
I mean, I guess the question is, what are we, what is our angle is not everything, right? Like, I think just to be clear that's like, we're trying to say, can we design the best chemistry to interfere with a model of what does it mean to be interfering with something? And you can sort of, one of the answers is about desensitization and long-term things is something that someone somewhere on this process needs to have a view on, right? I'm not sure it should be me or my company necessarily, but one of the things that we do think is that by like having new modes, you can interfere on the network in different places where it might be more or less likely that happens, right? And opening up those opportunities is where we see that being our place.
Hi there, I'm Matthew Fife. I'm from Storm Therapeutics in Cambridge, a biotech drug discovery company. I'd like to ask about the 60 billion. What was the 60 billion for? That wasn't Atomwise itself.
No, sorry, that's across the field.
Across
That
fields.
was like,
Okay,
the last 5 years last 7 years investment in AI for drug discovery.
Okay. And you're limited to enzymes, is that right?
So that depends on, like, so the platform is the thing that I described of doing transition state based drug discovery is all the technologies which say, for example, I talk about the pharmacophores, so that's finding new points that match based upon quantum detail rather than just sort of guessing it's a hydrogen bond donor. That is applicable to different things. At the minute, do we have the capacity to just take on any project? The answer is no. But does that technology allow us to do, say, QSAR in any problem? The answer is yes. In future, we're looking to prove that.
You said that by using this approach, you might be able to avoid resistance. Couldn't the residues involved in stabilizing the oxyanion hole, couldn't they be get mutated, and that lead to a mechanism for resistance?
I mean, yes, it's reducing the chance, it's not eliminating it. But if you look at sort of the studies that have been done in sort of, they're mostly in sort of antimicrobial resistance, or actually the best example is probably HIV, where all the protease inhibitors are could be argued to be transition state drug design. And that's in an environment where you see billions of variations of the sequence every day in a patient. You can get monotherapy to work for a really quite significantly long time. That's how sort of
But you still need to combine lots of different monotherapies
No.
to really get the viral level down.
but that's not true. Like in the
Mm.
sense that, like, if you look at in low income countries, they're taking lopinavir as a single
Single
therapy
agent.
treatment, right? And that's work. I mean, it's not perfect. It's not what you'd want to give people, but it, like, given this, that's like the harshest environment you could be. That level of evolution of resistance, it works pretty well. And if you look at sort of the 2000, 3000 generation of sort of malaria with these type of drugs, that doesn't show resistance. It's not impossible. It's just harder, right? I'm not trying to claim, again, we're not wizards. It's still just a.
Can I just ask about the final big? I've got a big picture question I just want to ask, and this is about without understanding communication is impossible. I'm a medicinal chemist. You guys are computational. Tim's got a medicinal chemistry background, but some of the questions I asked were about biology and getting the right target right in the first place. So target biology is so important, but these things are so complicated. How can we get together to discuss from different specialities in that sweet spot in the interface between all these? What is your recommendation?
I mean, I think the answer is working hard with people from the, like being in those groups, talking. I think it's quite boring. And I think it's also, I mean, we need to try different models of that. I think so. I've been in the academic space of trying to do that for a really long time. And it's always a struggle. The advantage being in the company is we've failed faster, I guess, and then iterated quicker on trying to find ways that do work.
Hi, thanks, David. My name's David as well, David Brown. That was a really nice talk. It's quite a naive question, but you talked about the challenge of communicating the science and all of that, and it seems like you have a really good control where you land on the same blind target that they found through a completely independent method. But the way that you present, you're using the quantum computing to find that oxyanion hole, and then you just so happen to have found the same drug that was already discovered a different way, right? Does it work the other way around if you just take drugs that we already know work pretty well? How many of them can you validate with the quantum computing? Is there the bandwidth to even try that, or are you forced to go novelty because you're a company?
I mean, the answer is we're forced to do the novelty, and if we had, I mean, the Atomwise's paper for their screening thing is what, 330, I think, studies. That takes a lot more money than we had to go and retrospectively do this type of analysis. And I think the scientist in me would love to do that on everything, and I think that that's kind of part of the future, certainly for the field, right? I don't know if our company is the right vehicle for it. And I think some of it, I mean, like, the other danger is that, like, it's so obvious. Like, if you try and replicate papers that you find out that that you knew what the answer was at the beginning of the study. And I think that's where we're hoping to do more and more blind trials to improve. It's the only way to improve it, right? And also to use data-driven methods to try and keep poking about where you're. And you phrased something that I just need to clear up, that the simulations are mostly quantum chemistry without the quantum computing. We use the quantum computing to inform decision making and setting them up, but we don't use the quantum computers to do it.
I'd like to follow up on that a little bit. You spoke a lot in your presentation about enzyme inhibition, and it seems like you've spent quite a lot of time there. I'm sure you guys have looked at a number of other neighboring areas where your methodologies could be applied. I was wondering if you could speak a little bit about that territory, that terrain, and what you see for the future.
So I think that the general principle of, like, so I mean, I think you've seen Schrödinger build a whole business over doing physics-based modeling alone and sort of trying, and I guess in a different way to Atomwise, replace screening and make your cascade faster and more agile in some sense. And I think there's no reason why the physics-based sort of abstractions we build can't be applied to all of these new modalities and actually help you in some ways. So, I mean, Tim is an expert previously in making PROTACs, for example, which is a modality I am slightly skeptical of, but like the only way to understand how do you take two different ends that interact with two different targets, bridge them, and get all of the properties correct is to understand when they're starting to interact with each other, right? And that's the sort of thing that naturally falls out of some of our modeling pathways. And So if we're going to ever do that in a rational fashion rather than just a sort of serendipitous one, then I think that works. I think there's a lot of this stuff whereby receptors and sort of just sticking chemistry where you want to break something. If you know one thing that does that, but you want it to be something that's useful in terms of like a drug, you're trying to optimize the properties of bioavailability or something. And that's the same problem no matter what you're trying to do, with some bells and whistles, which are that a transition state is more polar than another thing, but it's just a chemical detail.
Do we have any other questions on the audience? No.
So a transition state is relatively small in comparison to a protac ternary complex where you've got two big. So is the computing power there yet to look at protac ternary complexes?
It is there to look at them. The reason why we wouldn't start at them is for that, it's much more expensive to find out whether it's working, right? Although, in terms of going, if I have a crude model, can I work out whether or not the chemistry at this end is infecting the chemistry at that end? That is doable now. And there's lots of work about doing. I mean, it's the same in some ways. Like you think of it as a sort of protein docking problem plus, right? And I think that's where people can do that. Can we add value to that in the same way that we can add value to standard docking? I would say yes, but whether or not, because it's if you add, it's a like n squared problem, right? You need just keep scaling, right? And that's where it's still hard.
And when do you think? So you had the number of qubits increasing by a factor of 3 every year. When will we have enough qubits to do complicated drug discovery simulations?
Sorry,
2035 or?
So I don't think that people are going to have everyday access to quantum computers for that period of time or longer, maybe. But I think that being able to use niche applications to drive that sort of frontier forward of our understanding, I'm increasingly confident of the 5 to 10 year horizon of having useful things for niches.
Okay. Thank you.
I'm curious a little bit about that pushback. There are obviously limits in computing, and you guys are pushing those very actively. But are you also finding limits in how the theory is applied?
So I guess the answer is always yes, or at least like what you take from the theory and what you would do with it, right? So if everyone assumes, so the example there was this many ion-centered system, everyone's assumed that it's too hard to bother doing anything with. People have obviously not got a great understanding of the system if it's not that hard to deal with. And that kind of, those limits that way, but also like you find increasingly systems where you go, well, it just doesn't converge. No matter how high a resolution I do, no matter what I try and do to wrangle this system into a computable state, it doesn't work, which means there's something wrong. And that's at the highest level of theory, but actually like the more day-to-day area where we see this is that, so it's actually like, so I mentioned briefly and certainly had a conversation before this about how GPU accelerators allowed us to do molecular dynamics. So that's the kind of balls and sprigs physics version of understanding enzyme motion, for example. How when they came online, by 10 to 100 times the amount of simulation you could do, you suddenly realized that the potentials you used to parameterize them did not capture things, but just everything fell apart, or the temperature dependence was completely wrong. And so by doing that, sort of seeing those models, and we're seeing the same things again and again every time we run something longer or in more detail, that you see those things where the easy, quick parameterizations, which sort of in some ways encode your understanding, they fall over. And we see that all the time.
And I'd like to zoom forward a little bit in the future where we have more compute power and more understanding. Are we going to start to see this having an impact on personalized medicine? Is that the frontier after, or is that the frontier now?
Tim said he was happy to answer this, so I have an answer, but I'll.
I can have a go. Yeah. I mean, personalized medicine is pretty broad, but in the aspect of thinking of mutations, as you mentioned before, for particular diseases, our platform has the capability of doing the simulations specific for particular mutations. We can model these, and if it's transition state, or if it's the binding pocket that's involved in it, that mutation is there, then we can actually develop personalized medicines in that way. I think the only caveat is that we're still kind of doing early discovery And we need to factor that into the commercial viability of investing that much compute power, that much resource into the I guess the national or the global impact of that mutation. I think personalized medicine, as I said, I think that's the scope at which we can impact. I think beyond that, going to more individual based work, I think it might be beyond the scope of what we do, but I'd be happy to.
It's more looking at the biological aspects of the patient, the genomic, the genome, the transcriptome of the patient. That different from this, which is more discovering the drug. I think personalized medicine is this would be a component of it, but not.
And I think that's, again, I mean, it comes back to what is your library of things that you have, but also, I guess, you only understand, like in some ways, you only probe what you can probe. And one of the things that's interesting about this is, so I think really about like stratified medicine rather than personalized medicine, because it was like 10 or 20 years ago, there's a lot of talk about these N=1 experiments and stuff. And I think that's clearly silliness. Whereas like if you can get groups of patients where you see they do differently, but I can't tell whether or not an intervention works, by having more tailored interventions to particular variants of things and doing that relatively cheaply. I mean, it's interesting to see that when there's a financial driver already, so like it's horrible, but like when it was hard to get fentanyl imported to the States, you found that places in China managed to find tiny modifications of fentanyl that's still fentanyl really quickly, right? And if you had the same drivers for being able to find these sort of things that probed or potentially solved your problems on a chemical level, and you had a correct stratification, which is about, I guess, where you're going, which is like this sort of diagnostic side has a massive influence over where you invest your money in trying to solve things and what you think you can solve. So yeah, I think that would be a very exciting future.
Well, I think I'd like to take a quick step back here and talk a little bit more about Kuano and the barriers that you're seeing in adoption of this within large drug companies or indeed smaller biotechs. What are the challenges you see in growth and adoption of this methodology?
I guess there's sort of a variety of classes. Like commercially, there's been a sort of crash post for a variety of reasons, but including that AI was so hyped, right? That there was just, there's all of the air has been sucked out of the room, people willing to do exciting, novel things because they're a bit burnt. I think that will come back over the time, but you have to reach a much higher barrier. So I think for us, that means taking things much closer to the clinic than 5 or 10 years ago, a company of our size or with our level of innovation would have been asked to do before finding. But also people are more kind of, I guess there's like, for us particularly, we sort of came up in that previous world in some senses. So we look to be able to say, but this is a generic solution for your things, but tailoring it to people's actual real world problems for each individual company. I mean, I think everyone has their own target. If it's not an enzyme, we have already sort of said that's not currently our focus. So that's sort of like tailoring. And I think that's the next step for us as a company to work out. I mean, I guess a lot of the questions here have been, can you use our technology to solve my problem, is like the behind that. And can we extract these things and make them accessible to people for the problems that they want to solve? Which I guess is every. In some ways, just sounds like I've just rediscovered business.
Great. Well, thank you very much. It's been really a pleasure hearing from you and hearing your take on the future discovery through Kuano's methodology. If we don't have any further questions from the audience, no? Great. Well, thank you very much for coming. I really appreciate it. David and Tim, everyone.